Interpretation of laboratory results, Unit 4, Part 2, Microbiology. Microbiology is the study of microscopic organisms, and we have general microbiology and clinical microbiology. And in the MLT program, in the medical lab tech program, the students not just take the general microbiology class offered by the biology department, but in the program we concentrate in clinical microbiology, meaning that we are looking specifically to the organisms that are associated with causing human diseases. Microbiology has various subdisciplines. We can divide it into bacteriology, and that is the specific study of bacteria. Virology or virology, that is the study of viruses. Mycology, that is the study of fungi. In parasitology, that is the study of parasites. It is very important for us to understand and to be aware that there are many microorganisms that are not considered pathogenic to humans. And many organisms live in and on the human body and do not cause diseases. And in some cases, they even help the host, us, our body, with different processes, like the bacteria that needs to be present in the large intestine, in the gut. That bacteria, when those organisms, and it's not just bacteria, it's also some viruses that are known to be part of the human body, and they are called normal flora or microbiota or microbiome. And that means that those organisms do not cause diseases to the human body. And in order for us to identify microorganisms in general, we need to use different stains because different microorganisms have different properties and different characteristics. And depending on the stain property of those elements of those organisms, then the identification can be possible. We can try to grow those organisms using different media cultures, meaning having some kind of environment where the organisms can grow, where the organisms like to be, at the temperature they like to be, with the nutrients or substances that they like to feed on. So different media cultures are used in the lab. We also need to be aware that not every organism can be grown in the laboratory. A lot of viruses and actually a lot of bacteria can be very fastidious and they don't grow. It's very hard to grow those organisms. Viruses have to use human cells. So in the clinical microbiology, we do not do viral growth. So we do not grow viruses in the lab, not in the clinical lab. There are some research labs and some other labs that do have the capability of growing viruses. We also use biochemicals, different biochemicals and immunological tests, meaning the presence, we look for the antibodies that the organisms make to help us identify those organisms. We can use some molecular testing, looking for specific DNA of the organism, different RNA, different mitochondrial components. So we can use parts of those organisms to help us identify them. Then we want to determine what is the best treatment, what is the best way to get rid of those organisms if they happen to infect the human body. So we use different antimicrobial tests to get the susceptibility to see which substance, which antiviral, antifungal, antibacterial, antiparasitic substance can get rid of those organisms. We need to be very much aware that standard precautions, and sometimes not just standard precautions, but standard precautions plus some other special precautions, precautions can be used or need to be used when trying to identify different microorganisms, and it all depends on on how pathogenic they are. Slide number 2 is very, very important because unless you're going to be working in the laboratory as a medical lab tech, then your role in the microbiology in this part of laboratory is specimen collection and processing. Because we have bacteria and some viruses and even some fungi as part of our normal flora, it is imperative, it is very, very important that we follow the specific guidelines that apply to the collection of the specimen that is needed to identify the organism that is causing the signs and symptoms of the patient. If the specimen is not collected properly, following aseptic or sterile techniques, if it is not collected and actually processed and transported in the right medium, at the right temperature, within a specific time frame, that will compromise the results. Remember that this bacteria can be pathogenic, but it is also very fragile. Those are living cells that if they are, for example, exposed to air, when you're looking for an anaerobic bacteria, meaning bacteria that likes to grow where there's very little oxygen, and you expose those bacteria, that specimen, to air in the room because it's not collected in the proper container, then nothing is going to grow. And that may be reported as no growth, meaning no infection, but we definitely know that couldn't correlate with the signs and symptoms of the patient. So specimen collection is very, very important. You want to get to the spot where most of the organism can be collected. If you are collecting a nasal swab, like for COVID-19, if you have gone through testing for COVID, you know that they stick this very, very thin swab up your nostrils, and they try to go all the way. You feel that they are poke your eye out because they go all the way to the sinuses, because the virus is likely to be way up in the sinus area, not in the superficial around the exit, around the superficial part of the nostrils. So we want to have good collection techniques. We want to avoid contamination. We want to use the proper tools, the proper kit associated with whatever organism we want to identify. In slide number 4, we have some important factors that can predispose us to infectious diseases. This goes hand in hand with what we were looking for when we were talking about immunology. Depending on the age of the patient, the very young and the very old populations are very susceptible to infections because their immune system, when they are very young, is not properly developed. When they are very old, it's already weakened because of age, and that makes a difference. So usually our at-risk populations are the very young and the very old. So we need to consider that when we are studying or trying to diagnose somebody with an infectious disorder. The health or the immune status of the patient, as you have been hearing with COVID-19, the people that are most at risk of having severe complications of the COVID infection are the patients with pre-existing health conditions. Their immune system, their body is already compromised with trying to work properly with an underlying condition like high blood pressure, diabetes, or maybe they are going with they have cancer and they are going through treatments that weakens the immune system. Maybe there are other underlying conditions that will weaken the immune system and that will make them more susceptible to infectious diseases. We also have to consider the type and source of the infection. There are some organisms that are more pathogenic than others, and we don't need a big exposure to them. With just a slight exposure, we can become infected ourselves. We need to know that if the organism has to go in through the right port of entry, if you want to call it like that. If it is an organism that travels through saliva and, like droplets from saliva in nasal secretions, then if you step on it, you're not going to get sick, because that is not a mode of entry. It is not the right source of infection for that organism. So we also have to consider that when we are trying to see which factors predispose the patient. And incorrect antimicrobial use. And that is when patients are given the antimicrobial, either antibiotics against bacteria, medicine against bacteria, antiviral medicine against viruses, antifungal medication, antiparasitic medication. But if they do not take it properly and for the prescribed length of time, if they stop using it or taking the medication because it's either too expensive or is too painful, or it causes side effects, it makes them nauseous, or it causes other side effects, the patients might stop using it. And that incorrect antimicrobial use can actually predispose the patient to having a more severe infection. And again, lifestyle is very important. We want to help our body to maintain health, and we do need to exercise. We do need to be mindful of the type of food that we ingest. It's not just junk food; it's just the quality and the quantity. The use of illegal drugs, and actually sometimes the actual use of medication, when we use the medication improperly, can also have an adverse effect. And stress is detrimental to our health, and that compromises our immune system, and that might predispose us to be more susceptible to infection. So whatever you can do to reduce stress, that is for you to decide what works for you, but we are supposed to be able to relax at some time. Stress is good for a little while. It gets us through a rough time. It gets us through some due dates, but then after that, we have to relax a little and let the body decompress. Otherwise, we're predisposing ourselves to infectious diseases. On slide number 5, you need to understand this. This is important, and I believe it might be a test question, because these are the steps that are used when we are trying to identify bacteria. So if a doctor suspects a bacterial infection, either in the urine or in a wound or in the eye, anywhere, if the doctor suspects that there's a bacterial infection and he orders a culture and sensitivity, then this is the process that is going to be involved in us identifying the likely bacterium that is causing the infection, and the most likely antibiotic that will stop the growth of the organism or actually kill the organism. So again, specimen collection is very, very important. Once is collected and is transported in the correct, proper way, then we'll get it to the lab, and in the lab, in the micro department, we're going to do a gram stain. We're going to try to identify the type of bacteria that it might be present in the specimen, so we can see whether they are rods or they are round bacteria, or they are more rod-like, or they are spiral, or whether they have special staining properties that will identify them as gram-positives and gram-negatives. Those broad characteristics can give the clinician an idea of the antibiotic therapy that he can or she can start the patient on, even before they have the actual results. Then once the gram stain is done, then the culture. We're going to culture the specimen. We're going to use different media, different plates. Like you have it on the right, you have a plate that is a biplate. And we're going to, again, use medium that has the substances that most bacteria like. So if we know that we're looking for a specific bacteria, let's say a deep wound from a patient, from a diabetic patient, those tend to be anaerobic bacteria. So we might include more another type of medium than just your regular media. Then we're going to use the biochemical testing. We're going to be just mixing the colonies of those bacteria with different reagents so that we can figure out which is the organism that is the most likely. After we have identified the organism, then we're going to do the antimicrobial susceptibility test. And this allows us to see the dose and the correct or the most likely antibiotic that will kill the organism. So that is very important. That step number 5, that's where the sensitivity out of the culture and sensitivity name that you constantly see in microbiology comes from. So, and then sometimes it not be enough to just do the regular susceptibility testing. We would have to do some more very specialized immunological or molecular tests to identify or to see the proper treatment for that patient that is based on the results. Slide number 6 is just a little introduction to parasites. Parasites are huge compared to bacteria, compared to viruses, definitely. And they are considered some of the oldest identified microorganisms. There's actually some records of parasitic infections from the type of the Egyptians. So they have been around for a very long time. The thing about parasites is that they can start minute, microscopic, and then they can grow and grow and grow and grow and actually be, yards and yards and yards long, like those parasites, the hookworms that feed off the nutrients that we ingest. So yeah, it can be pretty bad. Lab testing, we can do different testing depending on what we're looking for. Wet preps would be done in genital secretions, like vaginal secretions, or just genital secretions for men and women. And those are done for whenever we're looking for Trichomonas vaginalis, and they look very interesting under the microscope. The ones that you see on the right is the first picture on the right, Trichomonas. Those have been stained. But in wet mouse, when there's no stain, what would give them away is that the flagella that they have makes them move in circles. So they go round and round and round and round. So you are looking under the microscope, and because it's a wet prep, meaning that we actually took a little bit of the genital secretion and mix it with a little bit of saline, put a cover slip and put the slide under the microscope, then we can see them just rotate in circles. And we know that those are trichomonas. So if we're looking for pinworm, we might do the tape test. The pinworm likes to migrate all the way to the anus, and you can actually see them at the entrance of the anus, and you can use a piece of clear tape to collect samples, send them to the lab. It is very common in children. It is very common in crowded populations. They do come out at night, and they can go from the anus part of one person and then go into another person. So it can be transmitted like that. So it's in Spanish. There's, I don't know how many of you have ever heard a a commercial in the Spanish-speaking channels when they have problems with pinworm, that they usually have a public announcement, and they put like a cartoon type, announcement, and it says, if you are, si tiene comezón en la colita, uh, uh, get tested. If you feel itchy in the anus area, get tested. Go to the doctor. Tell your mom. But it's because the cartoon is pretty it's engaging. Well, at least I laugh every time I see the commercial. But that is very common. Those parasites can come out and they make you itchy. So there's also serological or immunological tests that can be used to detect parasites, and those are becoming more and more common because it's easier to find the immunoglobulins or the antibodies against specific parasites in the blood instead of trying to find them in the different secretions. We can also look for parasites in blood smears, just like if we were doing a differential. Babesia, malaria, Chagas, those samples of diseases that are caused by blood-borne parasites. We can look for gastrointestinal parasites in the stool. So the patient would provide a stool specimen. We would do special washing and special preparation, and then look at the specimens under the microscope to look for specific parasites. We can also do tissue culture, and that is not commonly done. And whenever it's done, it would be done in a more specialized area in histology. So that is not common in the clinical laboratory, but it's an option. Then mycology is the study of fungi, fungus singular, fungi plural. And fungi include mushrooms, molds and filamentous, and yeast. So yeast can be considered an opportunistic fungi. And we have yeast, especially women, in the genital area. So it's there, and it's kept in check by the normal flora. If there is a drop in the normal flora, then the yeast can take over, and that's when we develop yeast infections. So infections caused by a fungus are called mycosis. And again, fungi are considered opportunistic in pathogenic diseases, meaning that they are opportunistic when they take advantage of a weakened immune system. And yes, they can be pathogenic, meaning that they can be really bad. They don't have to wait for the immune system to be weak. If they get entrance into the body, they can cause nasty infections. We have different types of fungi. We have superficial or cutaneous fungi. We have subcutaneous and systemic. And the worst ones would be the systemic, because that is when it goes into the internal organs. And to get rid of fungal infection is very, very, very hard, because they tend to resist treatment. So let's try not to get fungal infections. The other pictures right here, slide 8, gives us some examples of fungal infections or mycoses. We have superficial, subcutaneous, and systemic, just so that you can get grossed out a little. And then slide number 10 gives us a little bit of information of the opportunistic yeast infection. They are very, very pretty to look under the microscope, but you definitely do not want to have this condition. It's very hard to get rid of. Okay, and it's very uncomfortable. And then the last slide is just a little bit more information about viruses. Again, we don't routinely grow them in the laboratory. We need cell cultures, meaning we need to use specific cells to infect or to present to the virus so that the virus can get into those cells and then start replicating. So it's not an easy process. And just a reminder, viruses don't just cause viral infections. They are known to be oncogenic. A lot of them have been associated with causing cancer. So that's it. I hope this helps. This is a very short PowerPoint to compensate for the very long one for chemistry. And I wish you the best in your quiz. Good luck.